This lecture is for special senses vision and the structure of the eye and the sense of vision. First off, I apologize for the way some of these slides look. As you can see here, there's some of the eyelashes, the words are a little bit disrupted. So what I'm going to do is I will post the slides themselves also with the notes that are already also posted on Blackboard, so you have both. So first, let's start with the, so first off, 70% of all sensory receptors are in the eyes. Each eye has over 1 million nerve fibers that go through the retina. And so the human eye goes through cephalization. The sense organs are at the front, and you have quite a bit of sensory information that is held in our eyes. So there's protection for the eyes, and most of the eye is enclosed in a bony orbit within the skull, and there's a cushion of fat that you maybe have that holds it in place, and you remember that from lab and dissecting the cow eye. There's a lot of extraneous tissue that holds the eye in place. Some accessory stuff on the outside, you have your eyebrow, right here on the eyebrow. Up here, you have eyelashes, the hair on the eyelids. There's the conjunctiva, which is a clear association. We'll see it in a second, the next slide, that actually connects the eyelids to the cornea. You have a lacrimal apparatus. Here's a lacrimal caruncle. The lacrimal apparatus is above the eye, and it provides fluid that goes through and washes over and cleans it out. And just some other important stuff. You can see this is the medial commissure. This is your lateral commissure with opening of the eyelid. You have the iris, the colored part of the eye. Pupil is the hole in the middle or the dark spot. The opening is called the palpebral fissure, the opening. And then you have your cornea, right? And this is right here at the edge of the eye is where the cornea associates and connects, merges with the conjunctiva. The white part of the eye is called the sclera. I think that covers everything on the outside, accessory stuff that are on the eyelids. Okay, so let's go on to the next page. All right, this is showing you an anterior aspect. All right, so this is top down. Right, this is the eye within the orbit. So you see the eyelids close, right in the front, eyelashes, and you see above here, here's a lacrimal gland, which provides fluid, which washes over the eye. And you see this little thing in here, that is the conjunctiva, which kind of helps protect. It's a membrane that lines the eyelids, it connects the outer surface of the eye, connects to the outer surface of the eye itself, and it secretes mucus to lubricate and keep the eye moist. Conjunctivitis is a bacterial infection of this membrane, also known as pink eye. Okay, the lacrimal gland itself, right, and the ducts that are associated with it, and then kind of drain through here and move fluid over. This is kind of where it looks like. Here's your lacrimal gland, ducts, fluid tears come out through there, wash over, and remove everything that's here, and then whatever else goes through this little hole here, and then down into the lateral canal, the lacrimal canal, or caniculus, a small canal. There's a little lacrimal sac, which right in the lacrimal bone of the skull sits right in there, and that duct comes down into the nasal cavity, and that's then eventually empties into the nasal cavity itself. Okay, the function of this is to protect, moisten, lubricate the eye. It's the lacrimal secretions or tears, and they contain mostly, it's a dilute salt solution, some mucus, some antibodies to help protect the eye, and also a substance called lysozyme. Right, lysozyme. So let's talk about a couple of the other accessory structures of the eye, the muscles of the eye. All right, the eye has basically 6 distinct muscles, which we'll see on the I'm sorry again for this slide, but you'll see it on the slides from here. You have a couple. You have a superior oblique muscle. Okay, it goes along the top, kind of goes like this. And you have an inferior oblique, and then you have a lateral rectus. You have an inferior rectus, a superior rectus, and then over on this side you would have a medial rectus muscle. Okay, and what they move the eye in a certain way, so to produce eye movements in an H pattern. Okay, left, right, up and down. Okay, and the oblique muscles are less important in that movement. You can see the axis up front. Okay, so you have your there's your trochlea, which is a ligament. You have the rectus muscle has been cut out. So you have a couple of the rectus muscles. You can see it in an H pattern that's there. Okay, each muscle, right? So lateral and medial move laterally and medially, and they are basically controlled by a couple cranial nerves: the abducens, the oculomotor, and the trochlear. Right. So the superior, right, depresses the eye, right, pushes it down and turns it medially. So when you kind of like cross your eyes and look down at your nose, that's the superior oblique muscle, and that's by the trochlear nerve. The other ones are ocular motor nerves. So when you are doing the cranial nerve tests where you're following pens, you are up, down, left, and right. You're checking, what you're doing is checking for the function of these cranial nerves and whether or not they're functioning properly. So now let's look at the actual structure of the eye itself. You have, basically there are 2 segments. There's an anterior segment and a posterior segment, which is in front of or behind the lens. Right, so let's draw a line through the lens here. Okay, draw the line through the lens, and back here, this is your posterior segment, and in front of the lens is the anterior segment. Okay, so we have this lens. So a couple different parts, right? So we have the ciliary body, which is a muscle that holds, the ciliary body, muscle that holds the lens in place. This is the lens. This is where the pupil would be, right? In this anterior segment, you have what's called aqueous humor. That's a watery substance that fills that portion, right? So we have the anterior segment, which is in here, right? So you have the cornea, which is over top. This is the iris. If you're looking at the iris right here, right, which opens and closes and constricts and dilates in order to increase or decrease the amount of light that comes through the eye through the lens. The ciliary body will actually just help distort the lens so that the image can be projected correctly on the retina in the back of the eye. The eyeball itself has 3 layers. You have a fibrous outer layer, which is the sclera, or the white of the eye. Underneath that, you have a vascular layer. That's in the middle. That's called the choroid. And then you have the retina, which is the we call the inside layer, or the sensory layer. Okay, so we have the sclera. The cornea is that clear, outlier, overlying, transparent. It allows light to pass through. It repairs easily, and it's the only tissue that can be transplanted from one human to another without fear of serious rejection. Okay. So then in the back of the retina you have a couple of here's your optic nerve, and you have a central artery that goes and provides blood vessels, blood and nutrients and vascularization to all of this, of the eye itself. And right here where that optic nerve comes in, those blood vessels comes in, is what we call the optic disc. Okay, that's an area, it's a blind spot where there are no retinal nervous cells. Right, the choroid is a blood-rich nutritive layer of the posterior eye, and then it has, in the anterior part, it's modified into that ciliary body and the iris. So the iris and the ciliary body are actually part of the choroid of this. So you see this, and I did this, I chose this picture on purpose because it has the white of the eye as the sclera, the red is the vascular layer, and that continues in the iris and the ciliary body, and then the yellow is the sensory layer. Okay. Here's another picture of this. This is actually a dissected eyeball. Right, so you have the sclera on the outside, the vitreous humor, which is a jelly-like, it's a jelly-like fluid. Right, it's thick. It helps prevent scattering of light. And then you have the rest of this, the aqueous humor in the anterior segment. Right, the margin of the pupil. Here's the lens, part of the ciliary body, and the iris that will distort the lens as necessary. And then the optic disc right there, and the output of the optic nerve. Okay. So the retina, we'll talk about the retina next, actually contains two layers here. I actually see it there. It's fovea central. It's an area next to the optic disc where there's just cones. There's no rods. There's just cones, and this is the area where you have the sharpest vision that's present in that area. Okay, no photoreceptors are at the optic disc or the blind spot. Okay, so we have the outer pigmented layer absorbs light. Okay, here's the pigmented layer of the retina, absorbs the light, and then you have rods and cones, which are important for the sensory receptor cells. Those are your photoreceptors, rods and cones. And then you have that's the inner neural layer. So this absorption of light in the inner neural layer that's there. And then you have these ganglion cells that are, so the signals pass from the photoreceptors, right, via two neuron chain, bipolar neurons, so there's these neurons, into the ganglion cells, right, and they leave the brain toward the optic nerve, right, and your optic disc is where you don't have any. So we have kind of like this. Okay. So rods and cones. So rods are most found towards the edges of the retina. They allow vision in dim light and peripheral vision, and all perception is in gray tones. So rods are gray tones. All right, while cones, which are these guys right here, cones allow for color vision. Okay, color vision. They're most dense in the center of the retina, and then they have this area which is the fovea centralis. Right, the fovea centralis, which we see cones color. There are actually three types of cones. Different cones are sensitive to different wavelengths of light. Let's see the structure of the retina, right? And there's actually you can see it a little bit better. Here's the optic disc, comes in, you have this part, here's the rods and cone, the pigmented layer absorbs light, and it will stimulate these rods and cones, which then transfer that to the optic nerve. Right, so we're actually going all the way to the back and then start moving back towards the anterior, and then those messages get pushed through the optic nerve and then deciphered as sight. All right, so let's talk about the rods. So you have different wavelengths of light, I think. So here you have 3 cones, blue, green, and red, and they're sensitive to different wavelengths of light. So you excite them differently. Okay, so that's the sensory layer, but light passes from the outside here, okay, through the pupil, through the lens, and actually gets flipped, and then it is absorbed on the retina. Okay, the lens itself is a hard, or a solid, semi-solid structure that can be distorted by the ciliary bodies in order to properly focus the image or the light that's coming and passing into the eyeball onto the retina correctly. Okay, when the lens becomes hard and opaque with age, that's called a cataract. Okay, vision becomes hazy, distorted, eventually can cause blindness. Risk factors for that include diabetes, frequent exposure to sunlight, heavy smoking, etc. There's a couple other things. Okay, so we went through the 2 chambers of the eye, the lens, anterior and posterior, and then the aqueous humor and the vitreous humor. The vitreous humor is the gel-like substance, right? So, that's what's found in here, right? The gel-like. And what it does, it actually helps to maintain what we call intraocular pressure within the eyeball, so that this eyeball doesn't collapse on itself. All right, so it helps maintain that. So if we undo all these drawings, all right, so now the pathway of light through the eye. All right, so here's a picture of a cataract. You can see how the lens is grayish color, and that distorts the light and on the inside of the retina, and you have hazy vision. Okay. And again, we've seen this picture, right? The pathway of light. Light must be focused. So here's your fovea centralis, the macula, and then blood vessels, optic disc. You see no stuff, and then you have the whole retina that's there, right? Lateral medial. So here's the pathway of light, right? So light must be focused to a point on the retina, so that for optimal vision. Light is bent or refracted by the cornea, right? So it's bent by the cornea, the aqueous humor, the lens, and then the vitreous humor in order to be focused on the retina optimally. The eye is actually set for an optimal distance of best vision about 20 feet or over 20 feet away. Okay, but when you focus things, you go through a process of what's called accommodation, right? That's the lens changes shape to focus on closer objects. So when you're reading, you're looking up and you are looking out in a distance more than 20 feet. Your eyes are focused at one thing, and then when you look down, you try to read, it takes a split second for your lens to accommodate, to focus to what you're looking at. Okay? So it kind of looks like this from a near source versus you can see how the lens is distorted to accommodate having the focal point of light on the retina. All right, the image that is produced is what we call a real image. Okay, there's a whole bunch of physics about the lens itself. The lens is what we call a biconcave disc. Biconvex disc. Okay, so it has convex means it's out, right? And this lens distorts this and makes it a little bit bigger and flips it. But you pick that up. It's a reverse from left to right. It's upside down, right? And it's a smaller than the original, obviously, right? So those are what real images are. And then your body, your brain, the differences are associated and integrated and deciphered by the brain, so you know kind of upside down images, or actually you see in the retina, are deciphered as being right side up. Okay, so the pathway of light through the eye, right? So it goes through the cornea. Actually, we'll type this out. So pathway, okay, goes through the cornea, then through the aqueous humor, then the pupil, the lens, then the vitreous humor, eventually ending on the retina. Okay, so you should know that as we go forward. Okay, so here's kind of where the visual fields. You have a fixation point. Both your eyes take in separate images. So if you cover one eye at one point in time, you still see a full field, but you might not see one particular area, right? So your eyes work, and they have this overlapping field here in the middle, right? So, and then you have this optic chiasm. So if you notice the tracks here, right? Both eyes, so your right eye has neurons that go to the visual cortex and the occipital lobe. Okay, on the left, so the right eye stays on the right side, but then there's also neurons that go over and innervate on the left hemisphere of the brain, so that the whole brain is being associated on either side, and you can integrate a whole image together with both sides. That's what we call optic tracks. Okay, and the point where so you have this optic track, so these neurons come over and they innervate on either side. Okay, and then you have, and the thalamus is important in that optic track, integrating those sensory information. And where they cross over is the optic chiasma. The optic nerves cross over. So the way that impulses from here, you have the optic nerve to the optic chiasma. Now it's on the optic track, it ends in the thalamus, and the thalamus integrates that. And there's what we call optic radiation. Those neurons that go to the back, to the visual cortex, the occipital lobe, the visual cortex of the occipital lobe in the cerebrum. Okay? And so finally, we have, sometimes there's disruptions in how the image is focused on the eye itself. All right, we have a couple of different things. So this is what we call emetropia. All right, eye focuses images incorrectly. All right, so emetropia is a correct image, right? So light comes in, lens focuses the eye at the right focal plane, you get sharp vision on the retina, everything's fine. But then you have two different points. So if the lens doesn't work, or does not curve the right way, or if the eyeball is a little longer than it's supposed to where the focal plane is, it doesn't match up in some way. So you see here the focal point is not on the retina. The focal point here is not on the retina. So myopic or myopia, right, is nearsightedness. Right, these nearsighted, distant objects seem blurry, and that's because the eye, the lens is not focusing the light at the sharpest point. All right, so it has a problem with that. All right, it usually results from an eyeball. It's too long, and then there's a correction. So you have glasses or contacts that will have a disrupted lens. It will distort the eye, and then your lens adjusts for that change, and then we have a correct focal point on the retina. And same thing for hyperopic or hyperopia. Farsightedness, right? Too short. Now the focal plane is actually beyond the retina, so you have fuzzy, too short, or from a lazy lens. So if the lens doesn't work, the ciliary body doesn't work as well and doesn't accommodate the light. So you have a convex lens, which will help further focus it, so the focal plane is on the right point. Okay, two other things that are important. We have a couple things, other parts of the eye. You have astigmatism, where you have images that are blurry, and it results from lights, the focusing as lines, not points on the retina, because there's an unequal or problem with curvature of the cornea of the lens. Somehow the curvature of the lens is disrupted, and it focuses, instead of having a point, you have these lines that are here. And then other imbalances or problems with the eye. You have night blindness and color blindness. Color blindness is a genetic condition that's due to lack of a type of cone. That's color blindness. And night blindness, inhibited rod function. Rods see in grayscale, right, in gray colors. So you can't see at night because you don't have enough carotene, and the rods aren't functioning very well. Okay, and the last thing is glaucoma. Glaucoma is a condition where you have increased pressure within the eye, the intraocular pressure, where that vitreous humor is. Glaucoma increases that pressure, and you can cause blindness by pushing pressure on the retina and disrupting the cells of the retina. So it's like pushing out. Imagine a kind of overfilling a water balloon to a point where it's really pressurized, and it's pushing on these cells of the retina and can cause those cells to die. So I will post these slides as well as the notes that go along with it. If you have any questions, email me.